Okay, good morning, everyone. Can you hear me okay? Yeah? Okay, great. So my name is Jeff. I'm a staff person in the center. I'm responsible for our imaging and computation core facility, and I work closely with Professor Murphy on the dynamic brain circuits cluster. And it's through that we've had most of our interactions with the CONP, the Canadian Open Neuroscience Platform. And this has supported a number of projects for graduate students and postdocs at the center, and you'll hear from 7 of them today. But before we get to that, I just wanted to make it clear this is a hybrid meeting, so we have participants joining us via Zoom. We have one speaker who's going to present his slides via Zoom, so we are being recorded, and this includes the questions. So the mics in front of you, keep them muted unless you're asking a question, and we want to hold the questions till after all the presentations have happened. When your mic is active, the camera will rotate to you and you will see yourself on the screen. So if you're in the Rudy North with us today, that's the plan for asking questions. If you're joining us on Zoom, then Nicole will be monitoring the chat and looking for raised hands, and she will bring your question to everyone's attention. So before we get on to the students' presentations, I just wanted to say a little bit about the Canadian Open Neuroscience Platform for those of you who are not maybe super familiar with it. This is a group that has the vision to propel Canada's basic clinical neuroscience communities into a new era of commonly shared, digitally integrated data and algorithmic-rich neuroscience research. So this sounds like something we're all interested in doing. And they are pushing this forward in a number of ways, including providing digital research infrastructure to promote the sharing of data and our open workflows. They're trying to build community by bringing scientists across the country together to think about this and work on this together. And they're supporting training of individuals, graduate students and postdocs through their Scholar Award program. And we're going to hear from 7 UBC awardees today. In terms of the digital infrastructure, this has come a long way, I think. So I've just put a snapshot here of the CONP's data portal. If you search UBC here, you're going to see the data that has already been contributed from labs at UBC. It's pretty easy to do this at this point if you create yourself an Open Science Framework project and tag it with Canadian Open Neuroscience Platform. They have crawlers that will automatically find it and ingest it into the portal and make it visible through this mechanism. So it's easy to do, and it's essentially ready for us to share our data in this way. In terms of our CONP scholars, these are the individuals you're going to hear from today. There's Ellen, Jen, Kaitlin, Nick, Abhijit, Hao, and Peter. So I'm not going to take up any more time. I'll invite Ellen up to get us started, and please hold your questions till the end.
Okay. Oh, okay, that's a lot louder. So my project centers around Huntington's disease, and this is a neurodegenerative disease that leads to movement, cognitive, and psychiatric disturbances. And the dorsal striatum is the main site of neurodegeneration in HD. And in my experiments, I'm using the technique fiber photometry to measure activity in the dorsal striatum of mouse models of Huntington's disease as they perform different behavioral tasks, such as the 2 shown here, the rotarod and the open field. So I'm using fiber photometry to measure activity of the GCamp sensor during these tasks. And so here's an example of fiber photometry in a mouse doing the accelerating rotarod. So this is a technique that allows us to measure fluorescent sensors in vivo during behavior. And so this mouse is doing the rotarod task at the same time as I'm looking at GCamp activity in the dorsal striatum. And here is just an example of an entire trial. So this is one mouse doing the rotarod while I'm measuring GCamp, and the area in red is when the mouse is on the rotarod compared to before and after. And we can also use photometry to measure other sensors. So I've also used it with the glutamate sensor GluSniffer to look at glutamate transmission during this task. And I'm also using Deep Lab Cut, which is a machine learning software to track the position of the mouse's paws during this behavior. So here's two examples. The top is a wild type mouse and the bottom is an HD mouse during the rotarod task. And I've used Deep Lab Cut to label the paws during this task to look at the paw kinematics. So the right side shows a graph of the vertical position of the right and left paws in blue and red over time on the task. So as the rotarod accelerates, you can see that the steps that the feet take get larger, and they also start slipping more where their feet go below the rotarod. And then the bottom mouse, which is the Huntington's mouse, has much more, their motor coordination is more impaired compared to the wild type mouse. And so using Deep Lab Cut and then followed by my own analysis in MATLAB, I've been able to extract measures such as the paw position, foot slips over time, paw height, variance in paw height, and you can do this over the course of a single trial or averaging this over days. And doing this, I was able to find some deficits in the Huntington model, YAP 128, including increased foot slips, as well as changes to the paw height, which actually worsens as the disease progresses and the mice age, as well, and also changes to deficits in the standard deviation of paw height, so the variation in their step size. And then I'm also looking at the open field behavior with Deep Lab Cut and MATLAB. So using this, I can look at standard measures such as total distance traveled and center time, and also more sophisticated analysis using machine learning software. So I've looked at different behaviors that the mice engage in over the course of the open field trial. And using B-Side, which is a machine learning tool developed by the ITRI lab at Carnegie Mellon, I was able to find differences in the types of behaviors that the Huntington's mice engage in the open field. And then for the GCAMP data itself, I've analyzed this in various ways. So we can calculate things like change in fluorescence, Z-score during different phases of a trial and compare those. We can look at specific GCAMP events and look at the peak analysis, so looking at frequency, amplitude, width of the GCAMP events. And we can also do something I'm kind of excited about, which is aligning data, the GCAMP data, to specific behaviors. So the bottom corner graph, I'm showing an example where I've looked at every time the mouse has a foot slip on the rotarod, and I've averaged the GCAMP activity around all of their foot slips for this mouse. And you can see that right after the foot slip occurs, there's this big peak in activity, which is when the mouse is now trying to regain its balance after slipping. And then I'm also doing other analysis with the coming soon with the open field, looking at specific behaviors. And what the GCAMP data looks like during those, as well as analysis of data with the red and green sensors together with photometry. And so all of this data will be available or already is available on these open source platforms shown here. So I'm sharing raw and partially analyzed data as well as protocols on the Open Science Framework, which I guess will then also be shared on the CONP platform eventually. I'm sharing my analysis scripts along with sample data on GitHub through the Dynamic Brain Circuits GitHub, and I'm uploading published data to the Dataverse. And I just wanted to mention some of the scripts and sample data that are available. So I have repositories for the rotarod behavior analysis, open field analysis, and for photometry analysis. And I also wanted to mention that these scripts can also be adapted to different behavioral tasks as well as different types of neural activity. And that's everything from me today. So I just wanted to thank everybody on this slide, as well as the funding sources, including the CONP, and thank you for listening.
Jen.
Hi, I'm Jen. I'm a PhD student in the Sierra Lab, and my research work focuses on parsing out the mechanisms that underlie microglia gene regulation in the brain. And microglia are the brain's resident immune cell. And during neurodevelopment, they must coordinate specific patterns of gene expression that are required for different things like migration, differentiation, and establishment of the homeostatic state in the brain. And this precise regulation is orchestrated by what's known as epigenetic mechanisms, which regulate and influence gene expression without actually changing the underlying genetic sequence. So as you can see here, there are a whole host of different epigenetic modifications. And with the advent of high throughput sequencing technologies, we can now profile these marks through various epigenomic sequencing techniques, including ATAC-seq, ChIP-seq, by cell sequencing, among many others. And unsurprisingly, there have been an increasing number of publications profiling these epigenetic marks in microglia gene expression, and just a few are shown here. So as these data sets are being generated, of course, there are data integration problems, and it becomes difficult to compare or replicate these findings because each research group analyzed the data sets differently, and they're also just currently only available in a raw data format that's pretty inaccessible to people who don't code or use bioinformatics. So consequently, there's a high underutilization of these key epigenetic data sets, which ultimately hinders the advancement of understanding microglial gene regulation. So the goal of this project, with the support of CONP and my lovely co-advisors, Dr. Annie Ciernia and Dr. Paul Pavlidis, I'm building an interactive web-based tool for analyzing and comparing gene expression and related epigenetic signatures in microglia across key time points in neurodevelopment. And this involves compiling both transcriptomics and epigenomics data from both our own studies, as well as some of the published studies that I showed you, into a sort of searchable database, which will ultimately allow users to navigate through these large-scale omics data sets without needing to have any coding skills. So my efforts in the first year of Microgliome have largely focused on data visualization specifically of reprocessed data sets from existing studies, which I've integrated into an R Shiny app. So Shiny is an open source R package that provides a structured framework for building web applications directly in R. So the reprocessed sequencing data was then wrapped into TrackViewer, which is an R package published fairly recently in 2019 for visualizing multi-omics data tracks into a layered and linear way. So I'll walk you through just some screenshots of the app that I built. So in the current prototype of Microgliome, users can simply click through the different data options available. So you can, for example, like type in your gene of interest, and you can look at combinations of data tracks across different studies. And you click Add Graph, you'll see a nice sort of layered visualization of that data pop up on your screen. And you can do this for up to 4 independent query-generated plots side by side, which is an important feature of Microgliome because it allows for direct and flexible comparisons of your genes of interest. So for example, you can examine your candidate gene of interest, let's say IL-6, for patterns of epigenomic regulation across different microglial studies. And the TrackView, our package integration, also allows you to export the plots that you generate so that you can have publication-ready figures right after your session. So under the About page, there is a brief intro to Microgliome and what it's all about, followed by specifics on each of the data sets that have been included. So this includes an abstract of the paper that the study or the data set comes from, what each of the tracks correlates to, and where on the geo repository you can find them. And all of the code for the Microgliome app, as well as for the data sets that were reprocessed, can be found in different repositories on the Ciernia Lab GitHub page. And eventually the protocols will be written up and posted onto OSF and other CONP related platforms. So for future directions, I hope to expand Microgliome to not only have data visualization, but incorporate additional data analyses features alongside the visualization. So things like being able to generate gene expression heat maps or perform enrichment analyses for different epigenetic marks of interest so that you can do some of the different functions listed here. And with that, I would like to thank DBC and DataBinge and the grad program and CBH community, as well as my lab mates and co-advisors.
Okay. Can everyone hear me? Good. Okay. Well, so my name is Kaitlin Sullivan, and I'm a PhD student in the Dombrowski lab. And today I'm going to be talking about some analysis packages that I created for mFISH during the past year as my time as a COMP scholar. So mFISH stands for multiplex fluorescent in situ hybridization. It's a spatial transcriptomic technique that I helped get up and running in the lab. Basically allows us to fluorescently tag mRNA present in a tissue section with single molecule and single cell resolution. The way it works is that we hybridize 12 probes to a tissue section, amplify, and then visualize those probes using cleavable fluorophores, imaging 4 at a time. So after 3 rounds of imaging, we have a full 12 mRNA visualized within a single section. Then we can dehybridize all 12 of those probes, add another 12 for 24, 36, 48, et cetera, gene targets within a single tissue section. So one of the analysis packages that I've created is a Fiji drop-down menu that basically takes all of the individual rounds of imaging, registers them to the same coordinate plane, then segments the individual cell bodies, quantifies the gene expression within each of the targets that we've looked at, and finally creates a nice qualitative overlay image that we can look at. Beyond this qualitative image, we also have the gene expression matrices that come out from the quantification portion. For this, I've created a Shiny app. This is just a screenshot of kind of what it looks like. Basically, with this Shiny app, we can visualize the data, as well as use machine learning techniques like dimensionality reduction and clustering to further analyze the data. So here, using data from our recently published eLife paper in the basolateral amygdala, this is the overlay image that comes out from the Fiji app. And then using the gene expression matrices, we can upload them into the Shiny app, look at raw gene expression, both the spatial location as well as in these dimensionally reduced embeddings for raw gene expression, as well as hierarchical clustering, where you can simply take a slider, select the number of clusters you would like, look at them again in space, as well as dimensionally reduced embedding. And so now this technique is being used a lot more frequently within our lab. And so I've had lots of alpha testing that has gone on through my various lab members. In another amygdalar nuclei, Rennie Kendrick has looked at the central amygdala. So this is what comes out of our Fiji app. And then you can just put it into the Shiny app and do your various dimensionality reduction and clustering. Also, another recently published data set in the Colloquium done by Brianna Bristow. Here's what comes out of the Fiji drop-down menu. And then this is what happens when you run it through the Shiny app. And also recently, Dr. Larissa Krauss has been using this for our human tissue experiments. So this is the human cortex using fresh tissue. And again, we can just run it through the app and quickly, without any coding, get all of your dimensionality reduction, clustering, and spatial information. And so hopefully we'll be moving on to sort of more beta testing soon, and this will all be available on GitHub as well as the Open Science Framework. And with that, I'd just like to thank my lab and the COMP for this opportunity. Thanks.
Hi, everyone. My name is Nick. I'm a PhD student in Tim Murphy's lab. And this project that I did with the COMP is called Meso-social. It's an open source approach to study mesoscale cortical circuit activity in mice during a social interaction. So we work with mesoscale imaging in the lab. This is imaging through
Nick?
the...
Sorry to
Yep.
interrupt. Can you just maybe speak a little bit louder?
Sure.
Okay,
Okay,
thanks.
how's this? Okay, so we're looking at mesoscale imaging in the lab. This is imaging through the skull. We're looking at GCamp activity. And so this is an example of what these fluorescence images look like. So we don't actually have single cell resolution with this technique, but we can make out these individual anatomical structures and the spatial patterns of activation associated with these structures. And so it's a nice non-invasive technique that we use, relatively non-invasive. So the goal of this project is to develop a simple and open source mesoscale imaging system that can be adapted to study social interactions in mice. And so to this end, we've developed a build guide with parts lists and costs for building your own Pi-based mesoscale imaging system. So this is an image of a Raspberry Pi, which is basically a small credit card-sized computer. Along with this, there's step-by-step assembly instructions for completing your own Pi-based imaging rig. This is all for under $3,000. To look at social interaction, we've adapted this rig to image the brains of 2 mice simultaneously while they're in close proximity. And we facilitate this interaction by mounting one of these rigs onto a rail, which is controlled by a stepper motor, which is controlled by the Pi. So we can control the distance between the mice and stage an interaction between the 2 mice in this way. So one of the first things we noticed when we were looking at this kind of data, just looking at the average signal across both brains here, we see that as we bring the mice together, there's the activity in both of these brains tends to become more synchronized. What I mean by that is you can see these large transients occurring kind of independently when they're separate. And then when we bring it together, when we bring the mice together, we see these large events occurring more in a correlated fashion. And so we measure the Pearson correlation coefficient between these 2 signals. And we see that in the group data, there's a much larger correlation between the 2 brains when they're together than when they're before they're in the period where they're separated before they're put together and after. And we also compared it with trial shuffled pairings. So during the together period, but with different mice. And we see there's no correlation here as well. So this kind of suggests that this correlation is more specific to the interaction than timing of events or something like that. We also looked at individual regions. So now we're looking at regions plotted on the surface of the cortex and correlating the signals between mice. And this correlation is now expressed in color on this matrix. And again, we can see a large increase almost across the board when they're together, but most notably a large increase in the posterior barrel cortex, which is the whisker-associated sensory cortex. So we're also measuring behaviors during these experiments. We've mounted a camera underneath the rigs, and we're measuring behaviors as basically as movements in these regions that we're highlighting on the video. And so these movements can look like this, and we get a movement vector, which we threshold, and then create a binary event vector for each of these locations. And then if we superimpose those behavior data back onto the brain data that we were just looking at, we see that a lot of these large transients actually occur coincidentally with these movements. And seeing that we saw that large correlation in the barrel cortex, looking at the whisker signal, we see that whisking initiates a large activation in the mouse brain, but it also initiates activation in the partner's brain. So moving forward, another interesting thing that we found was that it seems that the magnitude of this whisking response is dependent on the identity of the partner. So in our case, when we paired mice with cage mates versus non-cage mates, this is an average whisking response. The cage mate signal looked a lot larger. So we're hoping to investigate this in more detail using high-speed cameras to precisely track these whisking events, as well as linear models to more sophisticated method to decouple these behaviors and things like that. And we're also looking to move into more mechanistic things, looking at social motivation. So this is just a basic experiment with looking at sex preference. So this is a male mouse in the center, and you can see he's more interested in checking out the female stimulus mice. And so that's it for now. I just want to point out the UBC Brain Circuits GitHub. That's where you can find the parts list and build guide and code. Data is available on the FRDR and OSF. There's a preprint available on bioRxiv, and then this paper also has methods on the surgery. So finally, I just want to thank my colleagues, particularly Luis and Federico, and I want to thank the CONP and thank you all for listening.
Thanks, Nick.
Hey, hi. Is my voice audible? Okay. Hi, good morning. My name is Abhijit. So I'm a PhD student in the Woodward lab, and I'm going to be talking about behavioral CPCA. It's an open source multivariate analysis framework to uncover weak cognitive and psychological and physiological and psychological basis of mental health assessment questionnaires. Okay. Okay. The main aim over here is to uncover the links between mental health symptoms and cognition. So more specifically, we would like to find links between cognition as measured by various cognitive tests and the symptoms severity, which is usually measured using different questionnaire items. Usually in most of the analysis, like people do not use all the questionnaire items. What they do is they basically take a summary score, which is basically an average of all the questionnaire items, and use that in the analysis. Now, the main reason for using only a summary score or the recommended subscales is that people are afraid of getting false positive results because of multiple comparisons. So, but now there are obviously drawbacks like for this particular method. One of the main drawbacks is that you're assuming like uniform weightage to each of the questionnaire items by assuming a recommended subscale or by assuming equal weightage and by just taking an average and using a summary score. So now, and like the other drawback is you're not finding these optimal weightage or optimal sets of items which are linked to some other set of items. So here, if you are trying to link the symptom questionnaires onto cognitive measures, then there won't be any set of recommended subscales which will match all sorts of measures. So for each different set of measures that you're trying to link, there would be a different set of optimal scales which would match them. So, yeah, that's basically what the researchers are doing. So the solution that we are proposing is that we are going to use a supervised multivariate analysis on all the individual items, which will find the optimal set of items and scales which map both the sets of variables. Now, obviously, we will do rigorous reliability checks to make sure that there aren't any false positives, but also making sure that that there aren't any false negatives that you miss by taking summary scores. Okay, so the data that we are considering over here is a large international data set. It was collected across 11 centers and 7 different countries. And basically what the data set measured was, it measured 18 different measures from different cognitive experiments like source memory, dichotic listening, matrix reasoning, signal detection, etc. And it also took questionnaire items using 2 different scales. One of them is CAPS, which is the Cardiff Anomalous Perception Scale, and the Lonnie-Slade Hallucination Scale, which is the LSHS. So you basically have 48 items and 18 measures. Now we are trying to find a relationship between the 48 questionnaire items and the 18 measures. So the method that we are using is called constraint principal component analysis, which basically finds relationship between these two, or between any 2 sets of variables. Now, I'm not going to go into the math of the method. I think maybe if you guys are interested, we could have a chat about this offline. But intuitively, what CPCA does is that it basically finds components which optimally link both the measures, and that is what we are doing. Okay, so now let's look at the results. So as I said, the main aim over here is to basically link these 2 sets of variables. As you can see, that there are many different variables. Usually conventionally what has been done is people take a summary score across the questionnaire items, and you just have one particular variable over there, which is the summary score, and then you do a univariate analysis on that with each of the measures. So what we are doing is, instead we are doing a multivariate analysis, and what our analysis gives is this fine level association between the cognitive measures and the questionnaire items. So as you can see, each individual item is associated with other set of individual items in the other set of variables. Now, our method, which is an open source multivariate method, it obviously gives these very fine level links between these 2 measures. It also gives effect sizes for each of the associations and a corresponding significance value. So what's the main message over here? So the main message is that if you want to compare any 2 sets of variables, and one set of variables is they happen to have symptom questionnaire items, it is better to consider all the questionnaire items and not do a summary score, because by by considering all the items, you get this very fine level associations, which are impossible to get when you are using only a summary score. So all the links for the code are available on GitHub, and we also wrote a manuscript on this, which has been accepted in Cortex, but an open source version of that is also available on an open source archive server. Apart from that, what we are planning to do next is we are planning to make the code and the methodology available on the CONP portal. And apart from this particular data set, we are also using it on like many other different data sets. We are also doing simulation to compare this method with other methods. And that's it. Thank you. I would also like to thank CONP and my lab members. Yeah, thank you.
Good morning. I'm Peter Hogg. I'm a PhD student in the Haas lab. Today I'm going to be talking about an open source software project called DynaROI, which is a dynamic region of interest management software that I started developing a few months ago. And the talk's titled Universal Segmentation of Time Series Regions of Interest for Aligning Neural Activity to Morphology. So in our lab, we use two-photon microscopy. It's a type of fluorescent microscopy that is uniquely has advantages for in vivo imaging, allowing you to use fluorescent biosensors to measure neuronal activity within living animals to see better how neurons compute information by measuring proxies for neural activity like calcium levels using genetically encoded biosensors like GCamp, or using glue sniffer to measure neurotransmitter release across the neuron. But traditional forms of two-photon microscopy is very slow because it uses a technique of raster scanning. And this is where you use a two-photon laser to scan every single pixel in the imaging space, and then you lose a lot of time by dwelling the laser into areas where the region of the region you're imaging doesn't actually have any sample. So you may only have one neuron labeled, but you actually spend a lot of time dwelling the laser beam into dark space where you'd get no fluorescent photons from your sample. Because this is a very slow process and neural activity is very fast, there's a lot of advances in two-photon microscopy that are designed to speed up this process. These include techniques called like scan-lined angular projector microscopy, random access multiphoton microscopy, and two-photon holography. These techniques have a shared commonality that instead of sampling every single point in space with your laser beam, you actually use sample information to restrict where you excite your sample and then only image those areas of interest. So for example, in holography, you can shape the wavefront of your laser and then use beamlets to only excite areas of interest in a volume that are predetermined. Or with scan-lined angular projector microscopy, you can have predetermined regions of interest, which you then scan a laser beam opposed to a point, and then you can get the fluorescent changes of those regions of interest very, very quickly. So I'll use an example from our lab where we use random access multiphoton microscopy, kind of how this works. You first acquire a slow reference image volume, and then you find, for this volume contains a neuron. Then you select points of interest across the neuron's arbor that you want to measure, and then you use the random access multiphoton aspect of the microscope to only sample from those areas. The problem, though, is that these techniques often require user-defined positions or user-generated ROIs. And creating regions of interest is very slow, especially if you have a very complex structure that you're trying to predetermine areas to image from. So, and this can also lead to inconsistencies between users, because different users will have kind of biases in how they construct their ROIs. And also it makes it extremely difficult to image dynamic structures. So for example, a single brain cell early on in development, like the ones that we image in our lab, they have massive amounts of structural change. So this is a time-lapse image of a single neuron labeled with GFP, and the time interval is 5 minutes between volumes. You can see within minutes that parts of the arbor have extended, retracted, there's new protrusions occurring. And if you want to measure the neuronal activity in these dynamic structures, you have to be updating your regions of interest very, very quickly to use these fast forms of two-photon microscopy to get the neuronal activity. So the solution is to automatically generate ROIs during an image session and kind of to remove the human from the process of generating ROIs, and then to automatically register and update the ROIs over time during the imaging session. And then you can incorporate some post-hoc analysis about the spatial relationships between the ROIs. And that's where my open source software project kind of is trying to meet these solutions. So I started work on DynaROI, which has a pipeline where you take your reference image that you acquire using the slow raster scanning technique. You then perform pixel classification so that the pixels within your reference volume can then be put into discrete semantic categories. Then you can generate ROIs automatically based off of rules pertaining uniquely to each semantic category. And then you can repeat this throughout an experiment. You then use the ROIs during the experiment for the faster types of two-photon microscopy, either random access multiphoton or scanline angular projection microscopy. So the project's still in its infancy. I have some preliminary data where I've been able to successfully use a bit of a pixel classifier to determine what is and isn't neuronal. So I've been able to exclude a lot of autofluorescence, things like melanocytes within our imaging areas that that would otherwise you wouldn't want to image, and then to begin to generate regions of interest across an entire arbor. So the goal is to pretty much be able to create a very complicated structure within minutes, and it's very, very challenging to do that. So we're currently trying to replace the pixel classifier with a machine learning-based approach instead of an old school learning approach. I've rewritten the code for the ROI generation to improve the quality of that. I'm increasing the generalization of the pipeline so other labs can then use DynaROI to image their samples. I'm migrating the code to a Napari plugin to make it a little bit more user-friendly with a user interface based off of that environment. And then we're providing direct integration with ScanImage and Micromanager, so the ROIs generated can then be fed directly into microscope control softwares. So with that, I want to be able to thank my lab for their support for the project, my project supervisors, Kurt Haas and Kasper Pokorski, and the funding agencies, and particularly the Canadian Open Neuroscience Platform. So, yeah.
Thanks, Peter. So I hope that gave everyone just a quick overview of the level of sophistication of these projects and the willingness of everyone to make their code and data and just the outputs of their project open and available to everyone in the community. So let's thank all the speakers actually before we move on. And take questions. So if you want to ask a question and you're in the room, then press the button in front of you. And if you're joining us on Zoom, then Nicole will find you in the chat or with your raised hand. Anyone? Brian, go ahead.
This on? Okay, there's a lot of great talks there. The students did a great job. Just want to say that to start off, it's wonderful what they're doing. I wanted to ask a question of Nicholas. Just this is quite intriguing, this social interaction between the animals. I don't know where you are, Nicholas, but
Nick, are you still there?
oh yes, you're on the screen. Have you also considered maybe ultrasonics or olfactory cues between the mice? The whisking is intriguing, but just so I guess it's easy to expand this to other additional sensory modalities.
Yes, so we have looked at ultrasonic vocalizations in this experimental paradigm. We didn't see anything while they were head fixed, although we did see them when they're kind of relaxed in their own home cages. We're also currently setting up and running some olfactory experiments. So we just made an olfactometer system. So we're looking to look at things like identity and social recognition through olfactory cues.
Thanks.
Any other questions? Yeah, go ahead, Tim.
I guess first, wonderful talks by everybody, and reflects a lot of hard work, and the willingness to share these tools is really admirable. I should point out that the CONP, I believe, is going to have another scholar competition. I don't know, in the coming months. So we'll hear about that and look for messages. Let's see. I got a question to Kaitlin about the labeling of the neurons. So when you strip and then reprobe, do the cells ever look like different enough that you can't say this is the same neuron with a different, can you, I guess, do like have, how many labels can you say this is coming from the same cell for?
For the most part, the decover slipping, there can be like small like shear forces that cause like a little bit of warping, but usually we can pretty accurately identify the same region. And then the registration first does like a nonlinear, like takes the two images, registers them together, and then we use a nonlinear registration that basically like sort of makes up for any of the warping that could happen between the decover slippings.
I see. So then that's all on kind of a cell by cell basis.
Yeah.
Okay.
And anything that like, if like the Z-stack doesn't include, like from one round to another doesn't include a specific cell body, they won't be included. Only the cell bodies that are included across every round of imaging will be segmented and then analysis done.
More questions? Go ahead, James.
Yeah, really great talks, really interesting stuff. I had a quick question for Nick. A really cool data. Seems to me like this could also almost be sort of a crude form of mouse-to-mouse communication. I'm wondering about it. I guess does this depend on whisker touch? If you move them back so that they can see the whisker but they can't touch whiskers, do you still see this effect? And could you test whether you can, I guess, transmit different behavioral, or not behavioral, sort of emotional states between the mice? Like if you have a scared or perturbed mouse, could that be transmitted to the other mouse via this sort of whisking interaction?
So I don't know if you can, so to answer the last question first, I don't know if you can transfer that kind of emotional state through the whisking. I have seen papers that show through olfactory cues, a stressed mouse can make its partner stressed. And for for the whisking communication part and whether observing the whiskers moving activates the barrel cortex, I can't say that for sure. The reason why is because these whiskers move really fast, and the camera that we had been using did not have a high enough frame rate to really resolve them as precisely as we could in that last image where you saw those whiskers moving and touching. So I think we can redo those experiments and get a little bit more information about how these whiskers are actually moving and the brain signals associated with those. This was kind of just gross movement.
Any questions from Zoom, Nicole? No? Anything else on people's minds?
I guess I could ask a question for Jen. Look, it's like a beautiful tool and a nice opportunity to maybe compare across studies. I liked how you could kind of mine the data. Can you also compare, I guess, the same findings across, like, is it possible that, I mean, it's likely that some labs have done the same brain regions, the same probes, and if you do that, do you have to scale the data, or yeah, just, I mean, maybe comment on some of the challenges and opportunities there.
Yeah, so the goal is to ultimately be able to compare across data sets like you suggested, and different labs have looked at the same epigenetic marks in different contexts. So the goal is to be able to kind of click what you want to see and look at how those marks change for a specific gene of interest, or like in a disease model or different like experimental condition. But I think the caveat is that they have to be processed through our pipeline. So we would include a protocol in like OSF or on GitHub, it's already there for like if the user wanted to generate their own data and compare it with the stuff on Microgliome, they would have to process it in the same way so that the formats are the same and the different QC checks and all of that align. So that is one challenge of it, which is why we aim to sort of do the reprocessing for the user so that they can make those direct comparisons.
Any last questions? No? Okay. Well, I guess we can thank everyone again, and thank you all for coming to this first face-to-face / hybrid NRC session. And I guess just keep an eye out for the communications from the center, and it will tell us whether we'll be in this room or some of them will be on Zoom throughout the semester. Okay, thanks everyone.
I just wanted to add that this is the first time in 19 months that we've been able to have lectures in this room, and I can't emphasize enough the importance of trying to come in person, especially if we have an external speaker. It would be unfortunate if half the audience decides it's easier to come by Zoom. So thanks for coming today. Everybody is here. Encourage your lab mates to show up in person, and we can start interacting again. Thanks.